Hello, my lovely physiologists. Michele Glass here. We left off with our action potential, telling it like a story. We had, remember, team acetylcholine and team sodium, and they had like team A and team B. And then we had team potassium. And so our job here is to begin filling in those gaps and talking about it more like a process. And so to begin, we have our cell at resting membrane potential, which we can say is a negative 70 millivolts. This is determined by voltmeter across the membrane, and this is really due to the relationship with that sodium potassium pump. Remember, it has pumped 3 sodium out of the cell for every 2 potassium in, setting up 10 times more sodium outside of the cell compared to inside of the cell. When we talk about our cell at resting membrane potential, we should also think about like channels are closed. So here we're really thinking about the chemically gated channels or ligand gated channels, the voltage gated channels, and even depending on the type of cell, those mechanical gated channels, right? And so the next thing that happens is the stimulus. So the stimulus could be a sensation. It could be coming from another neuron, right? So the stimulus is talking about that signal coming into the cell. But here, what we're going to think of as that stimulus, this is that team acetylcholine. So acetylcholine is going to diffuse across the synaptic cleft, and it's going to bind to, I'm going to call them chemically gated sodium channels. Remember, we can also call these ligand gated channels. We can even call it a nicotinic receptor. So it's going to bind to those chemically gated sodium channels. And then the story I said, that's like acetylcholine is taking its seat, right? Or pushing the button. And this is binding to those channels. The channels open. And we have the initial flood of sodium into the cell. And here, this is where I called it the like team A, sodium team A, right? And here is an important term for us to remember. We have, this is our depolarization, but we're just in the initial depolarization, where depolarization, remember, is talking about movement of sodium into the cell, making the cell more positive until we reach what's called threshold. And I like to sort of think of my threshold as like my magic value, because at threshold, this is when we actually get the action potential to start. So what's happening at threshold is the cell is reaching a charge that opens up voltage gated sodium channels. And so in our OpenStax textbook, it tells us that the charge is negative 55 millivolts. In the Pearson textbook, it's telling us that its threshold is negative 60 millivolts. And so this number varies across cell types and across the literature then. And so let's kind of keep in mind threshold can be either negative 55 millivolts or negative 60 millivolts. And that way, no matter how it's worded in our materials, we're going to recognize this as threshold. And so threshold is when the cell membrane reaches this charge, and that's going to open up voltage gated sodium channels. That's going to open up voltage gated sodium channels. And so this is where we get team B, sodium team B. So we get, I'm going to put this in red because now we've actually started our action potential because here we have the depolarization phase of the action potential. So just in case I haven't been totally clear, what's going on here is the stimulus is opening up those chemically gated sodium channels, allowing for that initial flood of sodium. We call that like sodium team A. And remember, once sodium team A comes into the cell, that's going to lead to sodium team B, the rest of the sodium coming in. And so the rest of the sodium comes in because that initial flood of sodium depolarizes the cell to threshold. That initial sodium depolarizes the cell to threshold. And threshold, depending on your reference materials, will be either negative 55 millivolts or negative 60 millivolts. And the thing that's significant about threshold, the reason it's like the magic number or the magic moment, is because this opens up voltage gated sodium channels, allowing for all of the rest of the sodium to flood into the cell. And as the rest of the sodium floods into the cell, this depolarizes the cell. This is the action potential, right? So this is like now sodium team B is coming in. And that takes us to what we can describe as our peak value. And so our peak value is going to be plus 30 millivolts. So peak meaning this is as depolarized, this is as positive as the cell can possibly get. This is as positive as the cell can possibly get because two key events happen here. And think about them as simultaneous events. So it's not like number one and then number two, just two key simultaneous events. Voltage gated sodium channels close. It's more accurate to say they deactivate. So I'm going to go ahead and put in that term here. And then the voltage gated potassium channels open, or I'm going to go ahead and say activate. And so what this means is if the voltage gated sodium channels deactivate, sodium is no longer flooding into the cell. And so this stops the depolarization. And if voltage gated potassium channels are active, what that means is potassium leaves the cell, and this actually initiates repolarization, which is our next step that we see happening. So repolarization is the movement of potassium ion out of the cell, returning the cell to resting membrane potential. So just like we had a depolarization phase of the action potential, we have a repolarization phase of the action potential. And that's this whole period of time where the potassium is leaving the cell. You know, it's starting at a positive 30 millivolts, and then that charge drops until we get to negative 70 millivolts. Now, here's the key thing. Those potassium channels are kind of like slow to activate and slow to deactivate. Or maybe what you can think about is they're like a slow opening and a slow closing, right? And so what we see is that the cell, the neuron will actually hyperpolarize all the way to a negative 90 millivolts. And this is because potassium continues to exit. At that charge of negative 90 millivolts, we finally see the voltage gated potassium channels close or deactivate. This, y'all, is the action potential. So the action potential is from the moment of threshold, the depolarization to the peak level of 30, the repolarization towards resting, and then that hyperpolarization all the way to -90. This is what we mean when we say action potential. Okay. The final step that we have looking at here is the sodium potassium exchange pump. This is going to have the job of returning the cell to homeostasis. So remember, it's going to do 3 sodium out, 2 potassium in, setting up a high concentration of sodium ion in the ECF. We said 10 times as much sodium in the ECF compared to ICF, and setting up a high concentration of potassium ion in the ICF, or intracellular fluid. Okay, so this is the level of detail that we need to know. This is the level of detail that we need to know. So maybe watch it again, maybe read in your text, and then the next video, we're going to look at the action potential using a graph. Okay, so stay tuned for that one. As always, take care of yourselves and each other.